What really happened when life was born on Earth? Dear traveler, greetings. Today, we unveil the mysteries of a primitive planet swept by storms of fire and oceans of darkness. A raw world where the sky was poisoned, where the sea boiled with energy, and where the Earth's surface was still a chaos of rocks and volcanoes. And yet, amidst this incandescent turmoil, in the heart of the ocean depths, a quiet miracle occurred, the first spark of life. But before embarking on a new adventure, don't forget to like the video and subscribe to the channel so you don't miss anything. Thank you, and have a good journey. When we trace back to the origins of our planet, we discover a universe very different from today. It all starts about 4.54 billion years ago, when Earth is born from a vast cloud of gas and dust rotating around the young sun. At that time, the solar system teems with debris, fragments of rocks, and ice from the proto-planetary disk. Gradually, a denser mass attracts surrounding matter by gravity. This is how Earth, in its most primitive form, begins to take shape. In its beginnings, Earth resembled a ball of fire, entirely covered in magma. We speak of a magma ocean. It is an almost liquid expanse of rocks, heated to over 1,000 degrees Celsius. Nothing stable then, only lava, burning gases, and crushing pressure. No continents, no oceans in sight. Yet inside, the crucial process of differentiation begins. Heavy elements like iron sink toward the center to form the core, while lighter ones rise, constituting the Earth's mantle. In this molten chaos, Earth started to cool faster than one might imagine. Of course, it took time, but on a cosmic scale, the surface solidified quite rapidly. The mantle gradually stabilized, allowing the emergence of an initial Earth's crust. This progressive cooling paved the way for transformations essential to life. For a viable environment to appear, this furnace needed to be calmed, allowing water to remain liquid and creating conditions favorable to more complex chemical reactions. According to many scientists, it is between 4.41 and 4.10 billion years ago that the primary crust and hydrosphere started to form. The surface remained very hot, but much more stable. Thanks to the reduction of major impacts and strong volcanic activity, a primitive atmosphere gradually formed. Volcanoes released water vapor and carbon dioxide. Gradually, the vapor condensed, resulting in torrential rains. This is how the first seas were born, making the emergence of biological processes possible. It would be wrong to imagine a calm Earth at this stage. Despite the presence of liquid water and a more stable crust, the planet remains subjected to frequent meteoritic impacts. Some, very powerful, formed immense craters and locally vaporized water, disrupting the nascent atmosphere. Yet, in parallel, water continued to arrive, volcanoes released gases, and geological stability progressed. Gradually, areas capable of maintaining liquid water durably appeared. This period marks the birth of our first oceans, far more hostile than those we know today. Between 4.10 and 380 billion years ago, Earth went through an intense phase called the Late Heavy Bombardment. During this period, our planet, like its neighbors, suffered from violent impacts from meteorites, asteroids, and comets. Scientists have long debated its duration, but most agree today. A peak of collisions indeed occurred. It was likely caused by gravitational adjustments of the giant planets, notably Jupiter and Saturn, disturbing the orbits of many small bodies and sending them towards the inner planets. Despite the violent impacts, Earth already housed protected zones where liquid water, minerals, and energy converged. These reactive niches, like temporary lagoons or underground environments heated by geothermal energy, offered favorable conditions for prebiotic chemistry. Each collision could undoubtedly ravage the surface, but it also brought chemical elements, even complex molecules. These shocks may have stimulated the first organic reactions. Thus, in the heart of chaos, layer by layer, the foundations of the chemistry that originated life were laid. At this distant time, there were neither plants, animals, nor fungi. The forms of life, if they had already appeared, were likely limited to very simple microscopic structures, capable only of reproducing or maintaining a metabolism. No confirmed fossils date from this period. The geological record has been almost entirely erased over time. However, some ancient rocks present chemical or isotopic signatures, subtle clues suggesting that a living organism may have influenced their formation. Between 380 and 350 billion years ago, the first tangible evidence of life appears. No fossils, but isotopic traces of carbon suggesting a biological origin. Carbon exists in several isotopes, notably carbon-12 and carbon-13, which differ in their number of neutrons. Living beings prefer to incorporate carbon-12 into their molecules, leaving a particular signature in the rocks. When this signature is detected in ancient rocks, one can suspect biological activity during their formation. These discoveries come from sites like Isua and Aquileia in Greenland and Pilbara in Australia. The rocks of Isua and Aquileia date to about 3.8 billion years ago and contain isotopic evidence that may indicate the presence of primitive organisms. Pilbara, famous for its very ancient formations, also offers a glimpse into the geology and potential life of that time. Scientists analyze the proportions of carbon-12 and 13 in these rocks. An imbalance in these ratios, different from a non-living formation, is interpreted as a sign of past biological activity. These clues remain controversial because very ancient rocks have undergone profound transformations called metamorphism, which can alter their composition and blur the evidence. It is therefore difficult to confirm with absolute certainty that these isotopic signals come from life and not from geological processes. Yet, the majority of scientists lean towards the existence of primitive organisms. By combining these data, we suppose that life may have emerged before 3.8 billion years ago, in those niches where water, rock, and energy met. At that time, the early forms of life were likely prokaryotes, simple cells without a nucleus, long before the appearance of more complex eukaryotes. These microorganisms, resembling bacteria, might have lived in mineral-rich waters, drawing their energy from chemical reactions with the Earth's crust, or perhaps from the light of a young and less intense sun. Shallow or hydrothermal environments would have offered ideal conditions. However, these hypotheses remain uncertain, as direct evidence of these microforms is rare and subject to debate. These isotopic signatures are crucial. They suggest a living chemistry with a selective use of carbon. Back then, the Earth functioned as an immense natural laboratory, where oceans, rocks, shallow ponds, and volcanic vents served as chemical reactors. Under the effect of heat and other energies, water, carbon dioxide, and hydrogen interacted to create small organic molecules, which could evolve into complex compounds. These clues bring us back to the idea that these processes began very early, setting the foundations of an initial microbial ecosystem. At the end of this journey through time appears a cooled Earth, partially covered by primitive oceans, still shaken by cosmic impacts. This fragile balance might have been enough to create something unique, life. Without direct observations, our geological and chemical clues illuminate these uncertain beginnings, revealing the potential cradle of all current living beings. It is in this chaos of fire, water, and collisions that our history as a species seems to have taken root, marking the beginning of an extraordinary adventure. Between 4.2 and 3.8 billion years ago, a crucial stage marks the formation of the chemical bases necessary for life. It has nothing to do with the Earth of today, no trees, no marine creatures as we know them. Yet, it is during this period that prebiotic chemistry laid the foundations for future biology. These complex chemical processes set the stage for the emergence of the first forms of life, initiating a long journey towards the biodiversity that surrounds us today. At that time, Earth's atmosphere was very different from today's. It was mainly composed of carbon dioxide, nitrogen, and hydrogen, with small amounts of methane and hydrogen sulfide. Oxygen, a vital gas for most current organisms, was almost absent. This atmosphere is described as reducing or weakly oxidizing because it lacked almost all free oxygen. These particular conditions profoundly influenced the chemical reactions that prepared the ground for life. Imagine a sky without vegetation or oxygen production, an Earth swept by dust-laden winds, more mineral than blue. The storms were violent, the planet was still cooling, with extreme temperature variations. There was freezing cold at high altitudes, and on the ground, areas heated by the Earth's mantle, reaching temperatures of several hundred degrees. These conditions, although varied, remain hostile to any life as we know it today, but they promote complex chemical reactions essential for what follows. Despite this hostility, a true chemical kitchen was established. Indeed, the energy from lightning, ultraviolet rays, and meteorite impacts transformed the molecules of air and water. These reactions produced the building blocks of life, notably amino acids, essential components of proteins. These proteins would become indispensable for biological functions such as cell structure, transport, and catalysis of reactions. Thus, the complex chemistry, stimulated by the environment, prepared the ground for the emergence of life. To test the synthesis of amino acids, Stanley Miller and Harold Urey conducted experiments in the 1950s replicating the primitive atmosphere. They created a closed system containing carbon dioxide, methane, nitrogen, hydrogen, and hydrogen sulfide above a liquid simulating ancient oceans without free oxygen. Subjected to electrical discharges, heat, and radiation, this setup imitated lightning and sunlight, allowing the observation of the formation of essential organic molecules for life. After several days, the analysis of the setup revealed the presence of amino acids like glycine and alanine, essential for protein formation. This chemical soup demonstrated that under conditions similar to the primitive Earth, complex molecules could form spontaneously without external intervention. Although this does not directly prove the origin of life, this discovery revolutionized our understanding by showing that such prebiotic chemistry was plausible, paving the way for the progressive appearance of life on our planet billions of years ago. At the beginning, Earth was subjected to an intense rain of meteorites and interstellar dust, more frequent than today as the solar system was still stabilizing. These impacts, although violent, brought essential chemical packets. The hypothesis of exogenous delivery suggests that indispensable organic molecules, such as nucleic acids and some acids, also came from these celestial bodies. The analysis of current meteorites confirms the presence of amino acids, sugars, and carbon compounds, proving that these life's building blocks could be delivered to Earth, not just formed locally. Beyond meteorites, interstellar dust also plays a key role. These microscopic grains, present in cold molecular clouds, harbor ices and complex carbon compounds. During the formation of the solar system, some of this dust was incorporated into the nascent Earth. Thus, a significant portion of the organic matter necessary for life could come from these cosmic sediments deposited in the primitive oceans. This combination of energies and external contributions enriched terrestrial prebiotic chemistry. Despite a hostile environment, early Earth had vast mineral surfaces, notably pyrite, a shiny metallic mineral composed of iron and sulfur. These surfaces acted as catalysts, facilitating certain chemical reactions without being consumed. Organic molecules could adhere to them in thin films, promoting their interactions and the formation of more complex structures. Thus, these minerals help to accelerate prebiotic chemistry, preparing the ground for the emergence of life. Clays, particularly montmorillonite, play a key role in prebiotic chemistry. These fine-layered minerals trap water, amino acids, and other organic molecules. Their charged surface facilitates polymerization, that is, the assembly of small molecules into longer chains. For example, this enables the formation of peptides, short proteins capable of catalyzing other chemical reactions. Thus, clays act as a natural laboratory where molecules concentrate, interact, and evolve toward complex structures that prefigure life. One can also imagine small, shallow ponds where, with each cycle of evaporation and return of water, organic molecules concentrate in contact with minerals. These cycles promote polymerization, the formation of bonds between small molecules. Whether on clay or ferruginous minerals, these surfaces catalyze the progressive assembly of increasingly complex molecular building blocks. Thus, the succession of dry and wet phases plays an essential role in the chemical evolution toward life. At this stage, it is not yet a question of cells or organisms, but of fundamental chemical changes spanning millions of years. This period, between 4.2 and 3.8 billion years ago, follows the formation of the Earth by about 1 billion years. Even though this billion years seems long in geological terms, it is actually a short period that shows how quickly the earliest precursors of life could have appeared. Early Earth offered both the essential ingredients, gases, water, various minerals, and the necessary energy sources, such as lightning, ultraviolet rays, geothermal heat, and meteoritic impacts. These conditions favored the formation of a wide variety of organic compounds, including amino acids, key for proteins, as well as nucleic bases carrying genetic information. Gradually, carbon chains and molecular bridges formed, multiplying chemical interactions. Scientists estimate that the accumulation and progressive transformation of organic matter set the stage for the next steps. Prebiotic chemistry did not necessarily occur in a single cauldron, but rather in several different, sometimes distant environments. These basic building blocks then met or combined, transported by water, geological movements, or volcanic eruptions, promoting the assembly of the elements necessary for life. Each location contributes in a complementary way to providing a vast repertoire of organic molecules, thus increasing the likelihood of seeing more complex assemblies endure. On the other hand, this environmental variability promotes the robustness of chemical processes. If one type of reaction is less effective in one region, it may be more active in another region where the conditions of temperature, pH, or mineral composition differ slightly. The temperature varied widely on early Earth. Near hydrothermal vents, the water could reach several hundred degrees, sometimes more than today. At high altitudes or on the surface, it cooled to nearly 0 degrees. These strong variations created thermal gradients essential for prebiotic chemistry. Hot and cold water formed interfaces where molecules circulated, reorganized, and crossed steps towards increasing complexity, thus favoring the chemical evolution toward life. Mineral catalysis does not only accelerate reactions, it can also direct the assembly of molecules. For example, clay surfaces or pyrite crystals attract certain organic fragments, favoring precise alignments. These subtle interactions influence the final structure of molecular combinations. This role is crucial for the emergence of life because a particular spatial organization can initiate reproducible structures. Among many attempts, a lucky configuration could generate an autocatalytic chain, a chemical reaction capable of maintaining and repeating itself. At this stage, prebiotic chemistry still precedes any form of life. There are no cells, no membranes, no microscopic organisms. The living world, as we know it, will appear much later. But without this chemical foundation, the first cells, bacteria, and then all forms of life would never have appeared. Amino acids, nucleic bases, and other organic molecules are not alive, but they constitute the alphabet from which the biological history of Earth will be written. Today, many laboratories continue the study of prebiotic chemistry. The Miller-Urey experiments have evolved, integrating variables like different energy sources, varied atmospheres, or simulated volcanism. Researchers now detect more complex molecules, such as sugars and simple lipids, thanks to modern techniques. Furthermore, astronomical discoveries show that complex organic molecules also form in space and on other celestial bodies, reinforcing the idea that an exogenous contribution played a key role from the very beginning of Earth's history. Whether it's the surfaces of pyrite or clays, meteoritic impacts, lightning, or ultraviolet radiation, Earth offered multiple sources of organic matter. This diversity is a major asset. Life has a better chance of emerging when it has multiple paths to create its fundamental building blocks. Some molecules were born locally, others fell from the cosmic sky, or formed at the bottom of evaporating ponds. The result, an abundance of molecules ready to take the next step toward life. Prebiotic chemistry is not limited to amino acids. Primitive lipids, capable of forming membranes or droplets, also appeared, aided by minerals and energy sources. Phosphorylated molecules may have initiated the formation of nucleotides, bases of nucleic acids. Moreover, in the face of intense ultraviolet radiation, organic molecules could have protected themselves in rock cavities or under clay layers, offering them a refuge to survive, multiply, and evolve into more complex structures. These precursors, formed in various environments and then transported, eventually meet and interact, initiating increasingly efficient chemical cycles. The boundary between abiotic chemistry and dynamics close to living systems is thinning. After many attempts, nature creates a stable assembly capable of replicating, storing information, and using energy. Before that, closed systems like vesicles need to be formed to protect the reactions. For now, it is molecular diversity that is taking root. It is fascinating to see that even today, the chemical universe continues to perform spontaneous transformations. Yet, the modern atmosphere, rich in dioxygen, makes it difficult to exactly reproduce the processes of Earth's dawn. Through laboratory experiments, the study of meteorites and ancient sediments, scientists seek to reconstruct these primitive reactions. They analyze the influence of different energy sources, catalytic minerals, and the probable composition of the atmosphere. Gradually, chemistry and geology converge to offer a living image of ancient Earth. Gradually, the image of multifaceted prebiotic chemistry is emerging. The primitive atmosphere, the contribution of material from space, and the role of minerals that host and organize these first molecules. All of this sets the scene for a world still without life, but where the future of the living is being developed. This process spans hundreds of millions of years, giving way to randomness and diversity without any intention, only guided by natural laws. As the planet stabilizes after the period of violent bombardments, amino acids, nucleic bases, carboxylic acids, and other compounds proliferate, as much in the early oceans as in drying pools or caverns. Associated with volcanic activity and successive reactions, they weave an ever-growing complexity. This prebiotic chemistry, to synthesize it, is revealed to be both local and universal. Local, because it closely depends on the specific conditions of Earth at that time. Universal, because similar phenomena can occur anywhere in the universe where liquid water or other solvents, energy sources, and mineral substrates converge. The essential takeaway from this phase of Earth's history is that the formation of biological building blocks, like amino acids, is neither impossible nor miraculous. It fundamentally relies on elementary organic chemistry, activated by favorable conditions and a sufficiently long time to allow the emergence of complex combinations. Nothing is yet fully elucidated. Many unknowns remain about the exact proportions, specific locations, or chronological sequences. However, thanks to the analysis of ancient minerals, fossil rocks, meteorites, and laboratory experiments, research progresses. It gradually unveils how inert matter could organize itself to become what we call life. This transition occurred slowly over millions of years until it formed structures capable of maintaining themselves, evolving, and crossing the threshold into biology. Between 4.2 and 3.8 billion years ago, the primitive Earth witnessed a multitude of chemical reactions that forged the first building blocks of life. In the absence of fauna, flora, or complex biological structures, this silent phase laid the foundation for everything that followed. Amino acids, nascent RNA, small peptides, all prebiotic chemistry was activated. Gradually, more efficient molecular systems would emerge from these foundations. Life was not yet present, but already its promises were inscribed in matter. At the very beginning of its history, as the Earth was slowly cooling down and the first landforms were emerging, certain environments may have played a key role in the appearance of life. One of the great scientific questions is in what types of environments this life could have originated. To answer this, we must look at reactive microenvironments, restricted zones where complex chemical reactions could have occurred. In this narrative, we will explore 4 major candidates: evaporitic ponds, alkaline hydrothermal vents, acidic black smokers, and primordial ice pockets. Let's start with these shallow evaporitic ponds, also known as salt flats or temporary pools. On the primitive Earth, these arid zones formed after rains or floods before partially drying under the sun's effect. As the water evaporated, salts and chemical compounds concentrated. This cycle of wetting and drying favored chemical reactions. Molecules mixed, then concentrated, facilitating the emergence of complex structures. With the help of catalytic minerals like certain clays, these environments could have generated the first precursors of RNA or proteins. To fully understand these cycles of wetting and drying, imagine an arid landscape with days close to 104 degrees and cooler nights. The sun, less luminous than today but still rich in ultraviolet rays, quickly evaporated the water from the ponds, concentrating the molecules. Then, rain or dew dissolved everything again, favoring the creation of more complex chemical bonds. Even small waves or a light breeze could provoke reactions. This type of phenomenon is still observed in some salt marshes, true natural laboratories of prebiotic chemistry. Next to the evaporitic ponds, another key environment for the origin of life is found at the ocean's depths, along the ridges. These are the alkaline hydrothermal vents, like those at the Lost City site. Their water, with a basic pH, escapes from zones where the ocean floor is fractured and where seawater contacts the Earth's mantle. There, a reaction called serpentinization transforms rock into minerals like serpentine while releasing dihydrogen and heat. These temperature and pH gradients then fuel potentially prebiotic chemical reactions. Hydrothermal vents of the Lost City type are home today to microbes capable of using dihydrogen and carbon dioxide to create their own organic matter. On early Earth, devoid of free oxygen, this type of metabolism, known as chemolithotrophic, likely played a key role. The porous walls of these chimneys formed true microreactors. They concentrated molecules, favored reactions, and stabilized complex structures. With temperatures ranging from 50 to 80 degrees Celsius, depending on the depth, these environments remained compatible with the formation of nascent organic molecules. Another key point in these sources is the pH gradient. The acidity difference between seawater and the escaping solution creates a kind of natural battery. As in our current cells, this disparity can provide the energy needed for biochemical reactions. The Lost City structures could thus have served as incubators for the very first metabolisms. The iron and nickel present in the walls also play a catalytic role, facilitating the formation of carbon bonds. Long before oxygen, these environments already offered a haven conducive to the chemistry of life. A bit further from the Lost City sources, one can find another type of underwater formation, acidic black smokers. Their color comes from the iron and sulfur-rich deposits they release into the water. Unlike alkaline sources, they develop in a low pH environment, hence more acidic. Despite these hostile conditions, they release large quantities of dissolved substances essential to the formation of prebiotic molecules. On the flanks of these structures, the temperature can drop below 80 degrees, creating zones more favorable to certain delicate organic reactions. Acidic black smokers generate micromineral pillars and fine membranes around the fluids they eject. These structures act as natural filters, letting some compounds pass through while concentrating others. Inside, temperature and concentration gradients create genuine microhabitats. The cavities formed by iron and sulfur-rich minerals favor chemical reactions, especially between warm and temperate zones. Thanks to this microporous architecture and a constant supply of energy and minerals, these environments may have allowed the emergence of complex molecules. Even today, microbes capable of surviving on hydrogen sulfide or dissolved metals can be found there. Moving on to primordial ice pockets. Although the primitive Earth is often imagined as a hot and volcanic planet, some cooling periods may have allowed the formation of vast ice caps. Inside, small bubbles of liquid water, heated by geothermal energy or pressure, offered a unique refuge. Protected from ultraviolet rays by the ice, molecules remained more stable. Even better, the ice concentrates substances. By freezing, it pushes some molecules into confined pockets, where chemical reactions can begin. In these ice pockets, the temperature hovered around 0 degrees, much colder than in other envisioned environments. But this cold had its advantages. It stabilized fragile chemical bonds, giving molecules more time to evolve. This can be compared to Lake Vostok, buried under kilometers of ice in Antarctica. Though essentially sterile, it shows that large bodies of water can persist in isolation. In such an environment, the ice filters harmful UV rays, and the slow diffusion increases the chances of reactions between molecules. It is possible that some of the microorganisms that appeared later on Earth originated from ancient ice-covered nurseries, as suggested by some researchers. These protected pockets might have allowed life to develop there before spreading when global conditions became more favorable. They offered a balance between protection and chemical potential, enough to initiate a precursor to metabolism. In the long term, this primitive metabolism could have paved the way for more complex processes, such as photosynthesis, and then the emergence of the first multicellular organisms. In summary, life might not have been born in a single place, but due to a combination of complementary environments. Over time, the primordial Earth experienced geological and climatic variations. Fissures, intermittent basins, and cooling periods multiplied the potential sites for prebiotic reactions. When we observe the current flora and fauna, we note that many organisms retain traits inherited from primordial environments. Some thermophilic bacteria still thrive in hot springs, with enzymes active beyond 80 degrees. Others, like archaea, thrive in highly acidic environments, similar to the conditions of black smokers. We also find halophiles in salt marshes and psychrophiles in polar lakes. This diversity shows how effective the first adaptations were, giving rise to systems capable of replicating and protecting themselves. This reinforces the idea of a plural and progressive origin of life. Cold can preserve fragile molecules. By freezing, water concentrates various compounds in its liquid channels. Thus, ice could have protected and stabilized important potential prebiotic stages. Ultimately, the question of the exact origin of life remains unanswered, and it is unlikely that a single place can be designated as its exclusive cradle. For researchers, this diversity of hypotheses is an opportunity, as it allows them to explore several paths and better understand the laws of prebiotic chemistry. Life could have emerged in multiple locations, then spread, carried by winds, rivers, or oceans. Here, we will compare several major scenarios that attempt to explain how such complex structures as the first cells could have appeared on the primitive Earth. Each approach highlights an essential element of life: RNA, peptides, lipid membranes, or even the possible role of viruses. We will clarify the concepts, explain the terms, and place these approaches in the grand narrative of evolution, while respecting their chronology and complementarity. Let's start with the idea of the RNA world. Ribonucleic acid, or RNA, is a molecule composed of chains of nucleotides, small units formed of a sugar called ribose, a nitrogenous base such as adenine or cytosine, and a phosphate group. In current cells, RNA serves as the link between DNA, the carrier of genetic information, and protein production. But some researchers believe that at the origin of life, RNA already played several key roles by itself before DNA and proteins gradually took over. The hypothesis of the RNA world is largely based on the discovery of remarkable molecules, ribozymes. These are RNA fragments capable of acting as enzymes, meaning they can catalyze chemical reactions. According to this scenario, some ribozymes may have had the ability to replicate without the help of proteins. Although this self-replication was rudimentary, it would have been enough to trigger a form of natural selection. The most efficient self-copying RNAs became more numerous, while others disappeared. This game of trials, errors, and adaptations would have, over millions of years, refined their structure and paved the way for increasingly complex functions. Of course, a question arises. How could the first RNA molecules form on a still hostile primitive Earth? According to researchers, the basic elements may have formed directly in the ancient atmosphere or been delivered by meteorites. Once concentrated in suitable environments, like small warm ponds or hydrothermal vents, these simple compounds could have gradually assembled. Evaporation, drying cycles, natural mixing, and the presence of catalytic minerals would have favored the formation of nucleotide chains. Laboratory experiments have attempted to reproduce these processes. Although nucleotide synthesis remains complex, several partial successes show that, under certain geochemical conditions, RNA fragments can indeed appear spontaneously. This theory presents an appealing advantage. It brings together in a single molecule the ability to store genetic information and to catalyze chemical reactions. However, it still raises several uncertainties. The precise steps of RNA synthesis under primitive Earth conditions remain poorly understood, and its stability in such an environment is far from assured. Despite these areas of uncertainty, the RNA world hypothesis remains one of the most studied. It offers a coherent bridge between prebiotic chemistry and the emergence of systems capable of heredity and evolution. The proteinoid world hypothesis suggests that the first actors of life were short peptides called proteinoids rather than ribonucleic acid. These chains of amino acids could have catalyzed certain chemical reactions and formed stable structures, playing a fundamental role in the early stages of biological organization. Even before the appearance of RNA molecules capable of catalyzing their own reactions, short chains of amino acids could have acted as simple catalysts. It is now known that some reduced protein structures can accelerate chemical reactions. According to the proteinoid world hypothesis, these peptides, originating from abiotic processes, thus, without the intervention of living organisms, would have accumulated in various environments, the edges of hot lakes, volcanic fissures, or rock cavities. With cycles of hydration and dehydration, they would have adopted three-dimensional shapes, favoring certain reactions, such as the concentration of substrates to form new molecules. In the laboratory, proteinoids can be obtained by heating mixtures of amino acids. They then assemble into microspheres, structures made of peptides similar to mini protocells. Although they do not contain genetic information and do not reproduce, these structures show that simple proteins can already possess aggregation and catalysis capabilities useful in a prebiotic environment. The proteinoid world hypothesis highlights the chemical flexibility of peptides and their ability to create microenvironments favorable to the emergence of life. But one question remains: how to transition from structures without a genetic code to cells capable of transmitting information. Despite this limitation, the theory opens up avenues on the multiple paths early life might have explored. For some researchers, the explanation lies between the RNA world and the proteinoid world. The co-emergence or co-evolution theory suggests that RNA and peptides evolved together, supporting each other from the start to create a more stable system. In the RNA-only scenario, its stabilization and replication pose challenges. In the proteinoid scenario, it is the absence of a reliable genetic code that raises questions. The co-emergence approach proposes that each compensated for the other's limitations. For example, short peptides can bind to RNA molecules, stabilize them, and help them maintain their structure. In return, RNA, with its molecular recognition capability, can direct the synthesis of specific peptides, thus laying the foundations for a primordial chemical collaboration. To illustrate this theory, biologists often cite the peptidyl transferase center, the functional core of ribosomes. These cellular machines manufacture proteins by reading messenger RNA and assembling amino acids. Their central core is made of RNA, and some of its loops called PTC interact directly with the amino acids. This structure suggests an ancient cooperation between RNA and peptides, with each element reinforcing the other from the beginnings of life. In this scenario, the goal is no longer to identify the first molecule that appeared, but to envision a joint progression. Peptides would have favored the first catalytic functions of RNA, while the latter would have guided their assembly or replication. Gradually, this collaboration would have led to more complex precellular entities, a prelude to the first cells. Often seen as a compromise, this theory is supported by various biochemical clues, even if experiments still struggle to reproduce all the stages of this co-emergence. Changing perspective, let's look at the fascinating world of megaviruses. When thinking of life, we imagine cells, but viruses, incapable of replicating on their own, occupy a unique position. Usually seen as parasites that appeared after cells, some researchers suggest that, on the contrary, giant viruses or megaviruses might be remnants of an ancient world. Their capsids, these shells housing the genetic material, could even have played a part in organizing the first living structures. Among these megaviruses are mimiviruses and pandoraviruses, discovered recently. Their genome, much larger than that of classical viruses, even contains genes associated with replication, repair, and translation functions absent in simple viruses. This singularity leads some researchers to think that these megaviruses could be the degenerate remnants of ancient cells. Conversely, they could be reconstructors, having contributed to evolution by exchanging genes with the first living cells. Let's imagine a primitive world where viruses were ubiquitous and very active. By penetrating rudimentary cells or protocells, they could have integrated new genes, modified their functioning, or inspired protection systems such as protein membranes. Although viruses depend on cells to replicate, some researchers support the virus-first idea. It suggests that fragments of viral code could have merged with primitive replicators to create more stable cells. This hypothesis challenges our understanding of life and remains largely unexplored. Now, let's explore the lipid-first theory, which assigns a central role to lipids. These molecules, integral to cellular membranes, form a barrier between the living and the outside world. Unlike RNA or proteins, they do not store information or catalyze reactions, but have a remarkable property. Indeed, they spontaneously form vesicles surrounded by a double lipid layer in an aqueous environment, similar to the membranes found in current cells. According to this theory, the spontaneous formation of lipid vesicles could have enabled the creation of a protosystem capable of housing and concentrating essential molecules, such as peptides or RNA. Depending on their nature and the environment, lipids organize into closed structures, micelles or vesicles, offering a major evolutionary advantage. These structures protect internal reactions, filter the exchange of nutrients and wastes, and promote the coexistence of molecules that would otherwise remain dispersed in the environment. The lipid-first theory suggests that these vesicles could divide under physical constraints, or even bud, when other molecules inserted into their membrane. If their interior contained a functional mix, like ribozymes, they could then serve as cellular precursors. Through competition, some vesicles would have gradually selected the most stable combinations of lipids and internal molecules, laying the foundations for evolution toward increasingly efficient structures. This model does not fully resolve the origin of the genetic code, but it clearly explains the formation of isolated compartments. According to lipid-first logic, the cell was first born from a protective envelope before its content organized into a system of chemical heredity. In other words, the membrane would have paved the way for the emergence of metabolism, thus crossing a decisive threshold toward the complexity of life. Let's adopt another perspective, the metabolism theory. First, it emphasizes the early appearance of metabolic pathways, these networks of reactions capable of transforming simple elements into more complex organic compounds while releasing or storing energy. According to this hypothesis, well before RNA or peptides, small autocatalytic chemical loops would have emerged, forming assembly lines capable of maintaining themselves, provided they had the right ingredients. A frequently cited example is the Wood-Ljungdahl cycle, a process observed in certain anaerobic microorganisms which live without oxygen. This cycle allows the transformation of carbon dioxide into simple organic molecules using hydrogen and iron and nickel-rich surfaces. On primitive Earth, geothermal zones precisely offered these conditions. This chemical cocktail could have enabled the emergence of reactions producing organic bricks, followed by more complex intermediates. It constitutes a pre-metabolism, long before the appearance of the first genetic molecules. The proponents of this theory argue that life is primarily a self-sustaining chemical system, capable of exchanging energy with its environment, much more than a mere information carrier. According to them, RNA or amino acids only appeared after the establishment of a metabolic core, producing the building blocks necessary for their synthesis. Everything would have originated from chemical reactions and not replication. This vision has inspired numerous experiments, notably around metallic minerals and hydrothermal environments, simulating the Archean. The major challenge remains to explain how a metabolism can sustain itself without a molecular code to guide or transmit it. Some researchers believe that the stability of minerals and the repetition of prolonged geochemical cycles would have allowed this self-organization until the emergence of molecules capable of conserving favorable evolutions. This perspective broadens the quest for the origins of life to mineral surfaces, energy flows, and primitive thermodynamics. Let's conclude this overview with the theory of panspermia. Unlike other scenarios centered on primitive Earth, this one suggests that the first elements of life could have come from space. In the lithopanspermia version, meteorites originating from Mars or other celestial bodies might have transported microorganisms or prebiotic molecules. Cometary panspermia focuses on comets, rich in ice and organic material. More speculative, the interstellar version imagines cosmic dust spreading life seeds from one system to another. We now know that some meteorites contain amino acids and other compounds related to RNA. Comets, on the other hand, house complex carbon molecules, showing that organic chemistry also exists in space. In the context of panspermia, these objects could have struck primitive Earth and deposited preformed materials. If rudimentary life existed elsewhere, it could have traveled in rocky fragments, shielded from radiation, and then reactivated on Earth as soon as conditions became favorable. Panspermia does not explain how life originated in the first place, because if it comes from elsewhere, the question simply shifts to space. But this scenario broadens our perspective, making the cosmos a vast reservoir where grains of life or pre-life travel between worlds. It questions Earth's place and suggests that life, in various forms, could exist elsewhere. This is the entire focus of exobiology, to search for signs of life beyond our planet. Numerous scenarios on the origin of life are inspired by studies of microorganisms living today in extreme environments. Think of those from underwater hydrothermal vents, where temperatures exceed 100 degrees Celsius, or hypersaline lakes, almost hostile to multicellular life. These life forms demonstrate incredible resilience and reveal the diversity of survival strategies. They provide a glimpse into possible habitats on primitive Earth, where early adaptation and selection mechanisms could have laid the groundwork for current biodiversity. By keeping an open mind, one better understands why science struggles to favor a single theory. Each model, from the RNA world to co-emergence with peptides, attempts to illuminate one facet of the mystery. The lipid first theory, or panspermia, add other perspectives. Research advances step by step, guided by experimentation, the confrontation of ideas and discoveries in extreme environments, on Earth and in space. For now, these theories form a rich tableau, fueled by the diversity of ideas accompanying our quest. While many questions remain open, this overview reminds us that nature excels at inventing pathways to complexity. By broadening our research, we may find decisive clues in the solar system or beyond. Meanwhile, these scenarios remind us that life, in all its diversity, is anchored in a chemistry that once began to interact, eventually leading to the emergence of the first organisms. Between 3.9 and 3.6 billion years ago, Earth was undergoing a crucial phase, with simple replicative entities evolving toward the protocell. The planet already offered complex chemical environments, rich in organic molecules and mineral surfaces. The nascent life likely appeared as small assemblies capable of copying their sequences, similar to polynucleotides. Replication errors introduced diversity and selection. Indeed, some mutations improved stability or reproduction, others did not. This competition favored adaptation and prepared the progressive emergence of the protocell. On the primitive Earth, nucleotides, less elaborate than today, could already store rudimentary information. Linked in chains, they sometimes adopted shapes that allowed them to copy themselves or assemble other molecules. A strand could thus serve as a template for its complementary. Copying errors introduced mutations, some improved the stability or the rate of replication, others reduced it. The most efficient ones ended up dominating the primitive chemical soup. Replicating a molecule requires a balance between precision and diversity. Too faithful, the copy limits innovation. Too approximate, it accumulates fatal errors. Favorable mutations, true engines of evolution, could modify the shape of a chain and offer it new chemical interactions. This early Darwinian selection, acting on rudimentary fragments of nucleic acids, produced increasingly efficient lineages of polynucleotides. Over time, these carriers of information assembled and interacted, heralding the complexity of ancestral RNA and preparing the emergence of increasingly sophisticated biological mechanisms. In parallel to the evolution of polynucleotides, lipid encapsulation took place. Lipids, with a hydrophilic head and a hydrophobic tail, spontaneously assemble into vesicles forming a protective membrane. On primitive Earth, these structures offered a refuge to replicating strands, filtering substances, retaining useful molecules, and protecting them from pH variations, toxic compounds, or extreme temperatures. They also concentrated nutrients, accelerating replication. Sensitive to physical changes, they could split into daughter vesicles, sometimes sharing the content of the mother vesicle. The fission of vesicles, triggered by local variations in temperature or salinity, already foreshadows cell division. When a vesicle, filled with replicating polynucleotides and newly integrated lipids, reaches a critical size, it becomes unstable and splits into two. Each daughter then inherits part of the molecules and resources necessary to continue the cycle. Repeated over long periods, this process allowed the preservation and transmission of a molecular heritage, while protecting these precursors from the environment and concentrating the ingredients to favor the synthesis of new molecules. Over time, the interior of the vesicles developed a proto-matrix reminiscent of the current cytoplasm, a mixture of water, metallic ions, and macromolecules. On primitive Earth, ions such as iron or magnesium stabilized chemical reactions by interacting with nucleic acids or simple peptide chains, creating specialized microzones. This local concentration favored polymerization, the stabilization of fragile structures, and pH regulation. By capturing these ions and accumulating cofactors, the vesicles became real microfactories where prebiotic chemistry and evolution enriched each other. To explain the nourishment of these replicative entities, the coupling between geochemistry and protobiology is evoked. In hydrothermal vents, hot water, rich in minerals, creates gradients of temperature and salinity. These differences, converted into chemiosmotic energy, rely on a proton imbalance across a membrane, as mitochondria and chloroplasts do today. On primitive Earth, lipid membranes could trap some ions, generating a slight proton motive force. This simple gradient was enough to trigger certain key reactions, supporting the synthesis and maintenance of essential molecules. Several billion years ago, Earth was a hostile but fascinating world where inorganic matter began to form new bonds. Forming continents, skies laden with dark clouds, and boiling oceans bore witness to a planet in flux. No plant, no animal, life was in its infancy. Yet, in this landscape devoid of fauna and flora, simple chemical processes would create the first building blocks of life. It was in this setting that the slow transition from abiotic matter to life began. To understand the transition between inert matter and life, one must first grasp the central role of carbon. Today, plants, animals, and microbes use it as the basis for their structures and vital functions. Several billion years ago, the primitive atmosphere, saturated with carbon dioxide from intense volcanic activity, was not yet familiar with photosynthesis. Other, more ancient chemical pathways devoid of cells began to exploit this gas. Natural reactions then transform mineral carbon into small organic molecules, the first building blocks of life's chemistry. Among the key reactions of this era is the carboxylation of pyruvate, where a carbon group from carbon dioxide is fixed onto this small compound, a true metabolic springboard. While it seems simple today, this step was a chemical feat at the time. It allowed for the formation of longer and more complex molecules, capable of fueling new synthetic pathways. In the primitive world, it could occur near hydrothermal vents, in contact with catalytic minerals, or in protected microenvironments, like under a thin rocky or icy layer. Another possible biochemical cycle is the reductive citric acid cycle, an inverse version of today's Krebs cycle. Instead of breaking down compounds, it progressively adds carbon and electrons to form richer organic intermediates. Under the conditions of the primitive Earth, this cycle could have functioned in such a manner. It presents a dual interest, providing key metabolites like malic or succinic acid and integrating carbon into a vast network of transformations, prefiguring the advanced metabolisms of modern cells. Following the first steps of life, we move from carbon dioxide assimilation to using other energy and electron sources. At the time, dihydrogen was among the major resources, released by volcanic activity and hydrothermal vents, rich in iron and sulfur. In contact with certain mineral surfaces or metallic catalysts, it could trigger reduction reactions, i.e., transfer electrons to other substances. This process allowed for the construction of more complex compounds or the transformation of molecules into elements useful for the development of primitive metabolism. At that time, protohydrogenases are often mentioned. Current hydrogenases are enzymes that catalyze reactions involving dihydrogen thanks to their protein structure and metallic cofactors. Before these elaborate forms, the primitive nature could have relied on simple surfaces rich in iron and sulfur, capable of manipulating electrons. These rudimentary assemblies favored essential redox reactions for metabolism. In hydrothermal environments, dihydrogen thus transferred its electrons to other compounds, initiating metabolic chains that would gradually complexify into biochemical networks. Sulfur also played a key role in early metabolisms. Present in various reactive forms, it could act as an electron donor or acceptor. Researchers mention an ancestral enzyme, thiosulfate reductase, capable of transforming this sulfur-oxygen compound into simpler forms, integrable into other metabolic pathways. Even today, some bacteria and archaea exploit this process to produce energy in extreme environments, like underwater hot springs or saline lakes. This rudimentary sulfur cycle already interacted with those of carbon and dihydrogen, laying the foundation for diverse microbial ecosystems. Alongside carbon fixation and the exploitation of dihydrogen and sulfur, a major advance emerged: the first pigments. These molecules absorb light at certain wavelengths and convert it into chemical energy. Long before plant photosynthesis, nature was already experimenting with simple pigment structures involving compounds like porphyrins or iron-sulfur complexes. Porphyrins are ring-shaped molecules capable of hosting a metal atom, as seen in the heme of hemoglobin or chlorophyll. In ancient times, they could spontaneously form from simple organic precursors, particularly in contact with iron-rich minerals. They would then capture light energy, which was more diffuse than today due to an atmosphere dense with dust and gases. This energy, even modest, could accelerate certain chemical reactions or promote the synthesis of carbon molecules, enriching the means of energy capture. Iron-sulfur complexes are assemblies of iron ions and sulfur atoms, forming small metallic nuclei within larger structures. They are still found today as essential cofactors for respiration and energy assimilation. In the primordial world, they facilitated electron transfers, allowing the reduction of certain molecules or the synthesis of organic compounds. Under the effect of light, they could also participate in photochemical reactions, channeling this energy to fuel chemical conversions useful to early prebiotic forms. The capture of diffuse solar light, associated with the use of dihydrogen and the reduction of sulfur, already sketched a primitive but varied metabolism where various chemical pathways reinforced each other. The carboxylation of pyruvate, or the reductive citric acid cycle, ensured the carbon base. Protohydrogenases and thiosulfate reductase provided energy and electrons. Finally, primitive pigments, porphyrins, and iron-sulfur complexes exploited light to intensify these reactions and broaden the array of available compounds. At this stage, the first forms of life, or proto-life, were neither structured bacteria nor autonomous organisms. They were chemical systems capable of sustaining chain reactions, transforming inorganic matter into progressively more complex organic molecules. Over repetitions and variations, these networks gained in robustness. Iron-sulfur cores excelled at electron transfer to synthesize new compounds. Meanwhile, mineral surfaces rich in iron and sulfur fixed small molecules and facilitated their assembly into more elaborate structures. These advances enabled the emergence of metabolic cycles. In certain environments, such as ocean floors or geothermal pools, abiotic chemistry gradually became biotic. Over time, reactions became more complex and were entrusted to more efficient enzyme precursors. Sites rich in warm water and minerals offered a natural laboratory, maintaining these processes active over long periods. Long before the appearance of plants and animals, these mechanisms for producing and managing energy laid the groundwork for future biodiversity. The first photosynthetic bacteria, which appeared much later, would exploit similar pigment systems to capture light and power complex electron chains, eventually enriching the atmosphere with dioxygen. This oxygen would then allow the rise of multicellular organisms, plants, and animals. But all traces back to these original reactions aimed at extracting chemical energy from the environment and storing it in carbon compounds. We can imagine these molecular assemblies drawing dihydrogen from volcanic sources, exploiting sulfur from minerals, and capturing the scarce photons passing through the primitive atmosphere. Initially rare and dispersed, these reactions would have concentrated in stable microenvironments, crust cavities or bubbles at the bottom of nascent oceans. Sheltered from cosmic radiation and fed by warm fumaroles, these chemical pockets accumulated sufficient precursors to multiply interactions and generate new molecular combinations. When the temperature remained compatible with the stability of organic molecules, generally below 100 degrees Celsius, these protosystems could persist long enough to pass through several chemical stages. At each stage, more stable or better adapted structures survived a little longer than the others. In this selective filter, more coordinated reactions appeared, aided by rudimentary cofactors like iron-sulfur complexes or porphyrin precursors. Without yet speaking of complete cellular organization, We can imagine microdroplets or basic lipid membranes encapsulating these small chemical factories. In this confined environment, the concentration of reactive molecules increased and the transfer of energy was facilitated. The proto-hydrogenases could then capture electrons from dihydrogen to direct them towards sulfur or carbon dioxide. Concurrently, the first porphyrins, fixed in the membrane or trapped in porous minerals, exploited light to stimulate the synthesis of certain compounds. Even without visible organisms, Earth was already animated by organized chemical dynamics. Abiotic processes gradually gave way to more structured schemes, combining carbon, sulfur, and dihydrogen in a growing network of reactions. The products of one pathway could serve in another, weaving a system of interdependent transformations. The first pigments, though rudimentary, complemented geothermal heat by capturing light, thus expanding the available energy sources in these primitive environments. For the fauna and flora that appeared much later, these chemical pioneers are the distant ancestors of all vital functions. Plants would descend from organisms that perfected the capture of light and the fixation of carbon through photosynthesis. Animals would inherit respiratory and digestive systems, extending the tradition of redox initiated by the proto-hydrogenases. Our food chain, down to its smallest links, thus plunges its roots into the metabolic capabilities forged in the shadow of a still inhospitable Earth. From a scientific perspective, it is fascinating to note the persistence of these principles. Our mitochondria, the energy centers of animal cells, still use similar mechanisms to transfer electrons derived from nutrients, relying on iron-sulfur complexes inherited from the past. Plants, on the other hand, depend on cofactors similar to the first porphyrins. This chemistry, billions of years old, remains embedded at the heart of modern cells, reminding us that our metabolisms extend an evolutionary history that began at the origins of life. Throughout this slow evolution, Earth established a dynamic equilibrium. Here dominated the chemistry of sulfur, there the exploitation of dihydrogen, elsewhere the discrete contribution of light energy. This rudimentary network foreshadowed the emergence of the first organisms capable of growth. The originality of these beginnings lies in the synergy between physical phenomena, hot fluid circulation, the diffusion of volcanic gases, and chemical reactions catalyzed by metals and minerals. Geochemistry and molecular biology show how crucial the geological context was. Without an abundance of iron, sulfur, CO2, and constant energy, Earth would have remained sterile. This era resembled a vast natural laboratory, where various geochemical niches favored distinct reactions. Near hydrothermal vents, sulfur-dominated; in evaporating pools, carbon-concentrated; in flooded caves, emerging porphyrins in contact with iron and subdued light transformed solar energy. Each site imposed its pH, temperature, and chemical orientation. Over millions of years, energy diversified. Gradually, the most stable chemical networks persisted, initiating primitive selection. The appearance of nucleic acids capable of storing information then paved the way for accelerated reproduction and evolution. We have just traced, in a simple chronological order, how modest reactions paved the way towards the concept of metabolism. Understanding what was at play then at the molecular level reveals natural ingenuity capable of transforming an inanimate planet into the cradle of evolutionary life. Slowly, these refined reactions gave rise to microscopic ecosystems and eventually to the diversity of modern forests, prairies, and living beings, all heirs to these ancestral metabolic choices. These chemical innovations paved the way for subsequent evolutions, transforming Earth into a living sanctuary where life spread and eventually shaped our current world. Thus, long before being confined within partitioned cells, metabolism already existed as a network of reactions shaped by the geological and cosmic conditions of the time. Modern living organisms retain a part of this heritage. Tracing this chemical path shows how much the history of life is linked to the properties of Earth, minerals, fluids, temperatures, and energy sources. If we discover life elsewhere one day, it is likely to start with comparable reactions of carbon fixation, redox reactions, and pigment production. In this narrative, we only have sparse fossil clues and analogies with modern metabolic pathways to guide our understanding. Yet, the chemical coherence is striking. The same logic of electron gain and loss, the same key metals like iron, and the same need to stabilize carbon in more energetic molecules. Although weak, sunlight provides valuable support to these mechanisms. It is in this bubbling crucible that the first milestones of what we call life were slowly laid down. Imagine traveling back in time, long before the emergence of animals, plants, and the landscapes we know. Earth is still devoid of forests. Its oceans contain no fish, and no beings tread its soils. The atmosphere and surfaces are transformed by powerful geological phenomena. In this primordial setting, very simple tiny organisms appear. Scientists believe that an ancestral group called LUCA, last universal common ancestor, gave birth to all current life forms. LUCA was not a single individual, but a population of microorganisms sharing the same genetic heritage. Understanding who it was and its environment means exploring the oldest root of our biological history. Researchers place this key period a little over 3.7 billion years ago, when fossils and rocks reveal the first reliable biological signatures. Some rocks retain isotopes typical of living metabolism, although their interpretation remains complex. Molecular clocks suggest that a common ancestor to all cellular life existed then. Between 4 and 3.8 billion years ago, terrestrial conditions were too extreme for sustainable communities. The partial stabilization of the climate and the crust allowed the emergence of stable lineages, including our universal common ancestor. To identify LUCA, researchers compared the genomes of primitive organisms, such as some bacteria and archaea, living in extreme environments. Advances in molecular biology revealed genes common to almost all lineages. Phylogeny indicates that LUCA possessed about 355 essential genes, including those for the ribosome, RNA polymerase, and chaperonins, responsible respectively for making proteins, transcribing genetic material, and properly folding new molecules. Each played a vital role, ensuring the minimal functioning of a primitive cell. The ribosome, a true protein machine, assembles chains of amino acids according to the instructions of messenger RNA, itself produced from the genes. RNA polymerase is the enzyme responsible for making these messengers. It reads the sequence of a DNA or RNA strand, depending on the organism, and synthesizes a complementary strand ready to be translated into proteins. As for chaperonins, they act as folding chambers, helping nascent proteins adopt their correct shape, essential for their proper functioning. All these systems indicate that LUCA already possessed an advanced protein synthesis machinery for such an ancient life form. To identify plausible LUCA genes, researchers conduct large-scale comparisons of DNA sequences from different families of organisms: bacteria living in hot springs, marine and saline lake, archaea, and even some more recent organisms. By identifying genes common to almost all these lineages and dating back very ancient times, they reconstruct a genealogical tree converging toward a central trunk. The more a gene is shared, the more likely its presence in LUCA. This approach is often associated with molecular dating, which measures the accumulation of mutations over time to paint a true genetic portrait of our common ancestor. LUCA, in addition to its genetic heritage, had a basic physical structure, a cell with a membrane separating its interior from the outside. Composed of branched fatty acids, it ensured internal stability by retaining vital molecules and regulating exchanges. This compartmentalization was essential. Indeed, without a barrier, the chemical reactions necessary for life would quickly dissipate into the environment, preventing any effective control. The hypothesis of a LUCA membrane composed of branched fatty acids is based on the diversity observed in the current microbial world. Bacteria primarily use linear fatty acids linked to glycerol by ester bonds, while archaea often employ isoprenoid chains linked by ether bonds. The branching could represent an intermediate stage, offering the flexibility necessary for survival in changing conditions, notably in hydrothermal environments. Stable and robust, this structure allowed for the preservation of cellular integrity in the face of abrupt changes in temperature, pressure, and chemistry. The study of LUCA's key genes and the geochemical clues contained in ancient rocks suggest an environment oscillating between 40 and 75 degrees Celsius, sometimes a bit more depending on the period. Moderately warm hydrothermal sources indeed offer nutrients and dissolved minerals favorable to life. The geochemical reactions occurring there, like serpentinization, release hydrogen, methane, and other energetic compounds for microorganisms. The water flow also creates temperature and pH gradients, favoring the formation of essential organic molecules. In such an environment, fragile nucleic acids have a better chance of remaining stable, allowing their replication and cellular survival. Gentle thermal gradients refer to the temperature variations over short distances, common in hydrothermal systems. For example, near a source, the water can reach 70 degrees. While just a few meters away, it drops to 30 or 40 degrees C. This gradual transition creates a diversity of conditions where microorganisms occupy their ideal zone. For LUCA, these gradients likely acted as a filter, selecting cells tolerant to moderate heat variations, while providing them with energy from chemical fluxes. They also facilitated nutrient diffusion, forming a microscopic ecosystem around the hot source. The genetic material of LUCA, whether RNA or a simplified form of DNA, had to remain vulnerable in a world sometimes reaching 75 degrees C. Moderate hydrothermal sources offered two advantages. First, they protected against extreme temperatures over 100 degrees, capable of degrading nucleic acids. Next, they concentrated minerals like clays, iron, or sulfur oxides, serving as supports where the material could adsorb, reducing its degradation and promoting its replication. Additionally, chaperonins present among the essential genes protected the proteins responsible for this replication. In this hydrothermal context, LUCA-like ancestors could exploit chemiosmotic energy. When the hot water of a source met colder water, it created ion concentration differences. Membranes, through specialized proteins, used these gradients to produce internal energy, foreshadowing the proton motive force of modern cells. The passage of protons through the membrane fueled the synthesis of energy molecules like ATP, essential for the production of amino acids, the replication of genetic material, and the functioning of chaperonins. Thus, thermal and chemical gradients ensured a continuous energy supply. Even if Lucca's hydrothermal environment was mild compared to other hotter sources, it remained unstable. Volcanic eruptions, variations in water flow, or geological changes could disrupt its living conditions. To survive, Lucca and its peers adapted their internal mechanisms, notably thanks to the branched fatty acids of their membrane, which maintained an ideal fluidity and avoided excessive rigidity or permeability. The essential genes found today in many extremophiles are the legacy of this long history of adaptation, at the origin of the current microbial diversity. Genetic and morphological data suggest that Lucca gave rise to two major lineages, bacteria and archaea. Over time, they acquired specific characteristics, particularly in their membrane composition, while retaining common genes. Certain enzymes, such as polymerases or ribosomal elements, remain very similar in both groups, indicating a shared origin. The main differences concern lipid linkages, cell wall composition, and certain energy metabolism pathways. Thus, from a simple ancestral cell emerged a tree of life, where each branch reflects a genetic gain or modification, facilitating adaptation. Contrary to popular belief, Lucca was not a rudimentary organism in the strict sense, even though it was simpler than current forms. Its estimated 355 genes already testify to an advanced organization, protein synthesis, genetic replication, and cellular protection mechanisms. We can imagine it as a robust microbe, resistant to moderate temperatures, modulating its membrane according to acidity, and exploiting the chemical resources of hydrothermal fluids. This sophistication was the culmination of billions of years of trial and error before stabilizing into a group of viable organisms, the result of long natural selection. With a functional protein synthesis system, organized genetic material, and a compartmentalized membrane, Lucca had the foundations to explore new metabolic pathways. Some descendants diversified their energy sources, while others formed communities around hydrothermal vents, exchanging nutrients and waste with symbiotic partners. Over generations, membranes and cell walls became more complex, allowing colonization of extreme environments. This adaptability stems from Lucca's founding genes, which enabled the biosphere to grow and diversify. Research on Lucca sheds light on the earliest history of terrestrial life. By tracing the genes shared by all domains, scientists have identified a common core, evidence of active replication, translation, and molecular protection mechanisms over 3.5 billion years ago. This universal ancestor is the result of the convergence of chemical, geological, and biological factors that allowed life to transition from simple molecular assemblies to sustainable and evolving systems. Lucca embodies the foundational act of our common tree, linking archaic microorganisms to the most complex life forms, including us. After Lucca, the first divergences between lineages led to new, sometimes radical, metabolic strategies that shaped the evolution of the biosphere. The resilience of life at that time suggests the existence of other niches, fostering diversification. Perhaps outside our hot springs, similar environments exist elsewhere, for example, on some icy moons of the solar system. The study of Lucca reveals that an elementary cell can already be a marvel of molecular engineering, exploiting thermal, chemical, and pH gradients. This adaptive ingenuity, carried by its key genes, remains the major legacy passed on to current life. At the time of Luca, fauna and flora as we conceive them did not yet exist. The biological landscape was dominated by microscopic colonies, bacterial mats, clumps of archaic cells, or primitive biofilms fixed on rocks near hydrothermal vents. By multiplying, these communities consumed dissolved chemical compounds and modified the local environment. The notions of fauna and flora only become relevant with the appearance of multicellular organisms. Yet, these early forms of life perfected replication and metabolism, laying the foundation for future kingdoms. By studying Luca, researchers are not merely satisfying a historical curiosity. They are exploring the universal nature of the processes leading to life. Understanding central mechanisms, conserved genes, and primordial membranes refines hypotheses about possible extraterrestrial life forms in similar environments. Warm hydrothermal systems, favorable minerals, and moderate energy gradients are found on other possible worlds, including in our galaxy. The quest for Luca thus guides exobiology and stimulates innovations to explore Earth and space. Advances in genomics and geochemistry provide an ever more precise portrait, reminding us of the value of life, even in its simplest forms. At the moment when the great story of life begins its branching, a major event occurs in the microscopic world: the divergence between bacteria and archaea. Far from being interchangeable, these two groups adopt distinct strategies to tackle the planet's chemical and physical challenges. We will explore their main characteristics, their lipid differences, their physiological innovations, and the impact of gene exchange on their rapid evolution. We focus on this key moment when two lineages separate and settle in primitive environments. Bacteria form a vast group of unicellular organisms found in almost all environments, from the human gut to hot springs exceeding 90 degrees to the icy waters of the poles. Archaea, long confused with them, actually constitute a distinct branch revealed by genetic analysis. They possess their own membranes and molecular machinery. In certain underwater hydrothermal vents, with extreme temperatures and pressures, they thrive with remarkable ease, demonstrating their extraordinary adaptability. To understand the importance of this divergence, one must first grasp the role of the cell membrane. It protects internal contents, regulates exchanges, manages energy, and perceives the environment. In bacteria, the membrane lipids are linked to glycerol by ester bonds, where an acid group attaches to an alcohol. In archaea, this link is ether type, attaching differently to the glycerol backbone. This nuance, far from anecdotal, strongly influences the thermal stability and resilience of the membrane in extreme environments. The ester bond, common in plants, animals, and many bacteria, makes membranes flexible. This is an advantage in various environments, but at high temperatures, they can lose effectiveness. The ether bonds of archaea, however, better withstand heat and pH variations, allowing them to thrive in extreme acidic or alkaline environments. Some live at over 100 degrees C, in fumaroles, hyperthermal ocean floors, or salt-saturated lakes, where few other life forms persist. Ether bonds also influence resistance to osmotic pressure, related to differences in salt or solute concentrations across the membrane. In salt marshes saturated with sodium chloride, where white deposits form on rocks, live halophile archaea. They are adapted to very salty environments. Their robust membrane limits molecule leakage and maintains stable functioning. Bacteria, however, rely on other chemical or genetic strategies, but are generally less effective in such conditions, illustrating how lipid composition reflects distinct lifestyles. Beyond membranes, the divergence between bacteria and archaea reveals the emergence of original energy strategies, including the sodium plus and hydrogen plus pump. Cellular activity often relies on ionic gradients. The cell expends energy to maintain different concentrations of sodium or hydrogen, which it then exploits to produce ATP, the primary energy currency of life. This pump, an active transport mechanism, circulates ions to create a controlled imbalance. This imbalance is then used by a molecular motor, like a watermill exploiting a level difference to generate usable energy. In nutrient-poor environments, being able to exploit a sodium gradient instead of hydrogen can determine survival. If the proton gradient is insufficient, the sodium flow speeds up metabolism. Conversely, in the presence of numerous protons and little sodium, some bacteria favor the proton gradient. Archaea, however, take advantage of this flexibility to colonize extreme habitats, from hypersaline lakes to hydrothermal vents, but also more ordinary environments like the intestines of ruminants. This sodium-dependent respiration, with its original ion pumps, constitutes another key marker distinguishing them from other life forms. To understand how bacteria and archaea have evolved so quickly, it's necessary to move beyond the idea of evolution relying solely on mutations and natural selection. They also practice horizontal gene transfer. Instead of transmitting their genetic material only to their offspring, they can exchange it with other cells, even unrelated ones. This common mechanism allows them to rapidly disseminate advantageous genes. Thus, a gene for heavy metal resistance, which appeared by mutation, can be passed on to another organism exposed to the same threat, making the boundaries between species much more permeable than in multicellular organisms, where such exchanges are rare. Horizontal gene transfers show that the divergence between bacteria and archaea does not imply isolated evolution. These regular exchanges introduce innovations from one group to another while preserving their distinct identities, marked by their membranes, metabolic pathways, and other fundamental traits. This dense network promotes mutual adaptation, an asset in facing ancient ecological crises, such as extreme temperature or salinity variations. Far from erasing their differences, these transfers have enriched their evolutionary paths and paved the way for a vast diversity of ecological specializations. From this rapid mixing emerges a vast pangenome, the set of genes that a group of microorganisms can share, even if each population expresses only a part of it. For example, in a volcanic lake, archaea adapted to high acidity, and others from a more moderately acidic area exchanged genes for proton resistance. This gives rise to new strains capable of occupying intermediate niches. This network evolves alongside morphology and physiology, continuously generating novel combinations. Bacteria also participate in this dynamic, accelerating adaptation through remarkable genetic fluidity. Since its beginnings, Earth has experienced profound climatic, chemical, and geological upheavals. This ability to exchange genes and modify membranes or metabolic pathways has allowed bacteria and archaea to withstand selective pressures. Coolings, intense volcanism, extreme temperature variations, glaciations, and changes in ocean composition have influenced salinity and access to energy sources. Every innovation, whether it concerns ion pumps or membrane structure, constituted an asset rapidly disseminated, explaining the persistence of these lineages, which still shape ecosystems and regulate various geochemical cycles today. In a scorching geothermal lake, mostly thermophilic archaea are observed, protected by their ether-linked membranes. In temperate environments, such as soils or roots, bacteria with ester linkages dominate, excelling in cooperating with plants, fixing nitrogen, and providing nutrients. Evolution has thus shaped a vast array of roles. In tropical forests, bacteria recycle organic matter, while in a desert geyser at over 80 degrees C, specialized archaea exploit energy from local chemical reactions. In marine environments, the distribution is similar. Bacteria dominate the microbial plankton, participating in the carbon and sulfur cycles. Some archaea live in oxygen-poor sediments, using hydrogen or various inorganic compounds. The sodium plus and hydrogen plus pump is crucial there. It allows some archaea to derive energy from the oxidation of hydrogen or methane, found in underground pockets or hydrothermal vents. Beneath the ocean's calm surface lies a range of microbial communities, each adapted to its own conditions of depth, salinity, and temperature. Long ignored, the dissimilarity between bacteria and archaea becomes clear through the study of their genetics. Archaea share with eukaryotes certain DNA replication and repair mechanisms while maintaining a wide diversity of metabolic pathways. This intermediate position intrigues researchers who question their possible role in the emergence of the first nucleated cells. This unresolved debate illustrates how the study of these two groups remains rich in insights into the origin and evolution of life. Today, the divergence between bacteria and archaea also results in distinct defense strategies. Archaea sometimes develop multiple cell envelopes, reinforced by protective protein layers against acidity or heat. Bacteria, on the other hand, often have a thick cell wall rich in peptidoglycans, an assembly of sugars and amino acids whose structure determines whether it is gram-positive or gram-negative. This criterion, essential in medicine, influences sensitivity to antibiotics targeting this wall. Archaea, lacking peptidoglycans, react differently. This has long confused their identification compared to bacteria and other known organisms. From a human health perspective, the divergence between bacteria and archaea also holds significance. Some methanogenic archaea present in our intestines break down dietary fibers and produce methane. Numerous bacteria colonize other areas of the body. Most are beneficial, but some become pathogenic under favorable conditions. This millennia-old cohabitation forms a complex microbiome, predominantly bacterial, but also including archaea. Understanding their differences sheds light on the impact of the microbiome on digestion, immunity, and even mood. Recent research shows that an imbalance in this system can have significant effects, confirming that the divergence between bacteria and archaea, once a matter for specialists, now has concrete medical implications. Why would life have followed two such different paths? Current hypotheses suggest that this divergence took advantage of the extreme conditions of early Earth. The first organisms specialized in hostile environments, exploiting resources inaccessible to others and thus escaping competition. Once these adaptations were established, they were passed on and enriched through intense gene exchanges. Gradually, these blocks of innovations shaped lineages capable of colonizing ocean floors, ammonia-saturated caves, or arid and corrosive lands. Later, bacteria found an advantage in symbiosis with plants and animals, facilitating the terrestrial expansion of vegetation. Archaea, meanwhile, retained their specialization in the extreme, occupying niches untouched by any other form of life. From a chemical standpoint, bacteria and archaea embody two creative responses to the challenges of the same planet. Bacteria have developed various forms of photosynthesis, the most famous of which is oxygenic photosynthesis. It releases oxygen from water and carbon dioxide. This innovation profoundly transformed Earth's atmosphere and influenced the global evolution of life. Archaea, on the other hand, exploit other pathways. Rather than light, they derive their energy from reactions involving methane, sulfur, or various inorganic compounds. This metabolic diversity has shaped ecosystems ranging from deep-sea environments to underground caves, lakes, and soils. Even today, fauna and flora depend on this complementarity. Some plants live in symbiosis with nitrogen-fixing bacteria, while herbivores rely on methanogenic archaea to digest cellulose. Through horizontal transfers, bacteria and archaea also exchange valuable molecular tools. Some key enzymes, useful for breaking down complex compounds or resisting toxins, circulate freely among populations cohabiting the same environment. This mechanism explains the rapid spread of an antibiotic resistance gene in a hospital when several microbial strains are in contact. In nature, these exchanges, although subtle, contribute to ecosystem balance. Indeed, a vulnerable strain can acquire resistance and colonize a niche previously inaccessible. Thus, the gene network reflects a web of shared skills, continually reorganized according to mutations and environmental conditions. This evolutionary panorama resembles less a linear path and more a vast network of interconnected routes. Junctions allow for gene transfers, modification of physiological traits, and exploration of new habitats. Bacteria and archaea have evolved in resonance, each following its own path, sometimes collaborating, sometimes competing to conquer hostile environments. This strategy enhances their resilience. In the face of a sudden change, some populations already possess, somewhere in this network, the genetic baggage or lipid structure required to survive and reproduce. This divergence manifests everywhere, including within us. In a simple sample of forest soil, thousands of species of bacteria and archaea interact with fungi, insects, and leaf litter. In the ocean, every gulp of water contains millions of microscopic cells, most of which remain unknown. Corals, seagrasses, mangroves, and large marine mammals depend on their metabolic activity. Whether it's the nitrogen or carbon cycles, the decomposition of organic matter, or the production of methane, the combined footprint of bacteria and archaea is omnipresent. Observing a botanical garden or a bacterial culture in a laboratory is to witness the legacy of this original divergence. Distinct membranes, innovative ion pumps, and rapid adaptation through horizontal transfers testify to the flexibility of prokaryotes. This heritage, rooted in terrestrial history, still supports the fertility of soils, the stability of ecosystems, and the existence of complex life forms like ours. Thus, the question, what differentiates bacteria and archaea, goes beyond membranes and pumps to touch every corner of the planet and fuel the flow of life. The time has come to delve into an enigma: megaviruses. These microscopic giants, still poorly understood, spark intense debates among evolution specialists. Are they mere remnants of a bygone era, or, on the contrary, evolutionary accelerators capable of disrupting and stimulating the emergence of new forms of cellular life? To understand this issue, let's look at their extraordinary size, enigmatic genes, and possible influence on the history of biodiversity. Their recent discovery explains the growing interest in their place in the tree of life. Let's take a brief step back in time. A few decades ago, viruses were described as tiny entities, devoid of any autonomous machinery, forced to parasitize animal, plant, or microbial cells to multiply. But in the early 21st century, the discovery of megaviruses, nicknamed the giraffes, shook this vision of the viral world. Among them, mimivirus and Pandora virus, whose capsid, the protein shell protecting their genetic material, is visible under a standard optical microscope, a rare fact for a virus. The first upheaval concerns their size. A virus was previously defined as a submicroscopic element, visible only under high magnification. Yet mimivirus measures several hundred nanometers in diameter, sometimes nearly a micron, making it visible with less magnification. Its capsid is impressive in its dimensions, but its genome is even more astonishing. While viruses were thought to be limited in genes, some megaviruses possess a repertoire richer than that of certain bacteria. To gauge the significance of this discovery, let's remember that the genome contains the instructions needed for the production of proteins, essential to any form of life, be it microbes, plants, or animals. Viruses, unable to replicate on their own, usually have to parasitize a host. Yet, in megaviruses, unexpected genes are found, including some related to protein synthesis. Among them are reduced versions of enzymes called aminoacyl tRNA synthetases. These enzymes attach an amino acid to a transfer RNA, the carrier that delivers the amino acid during protein assembly in the cell. That a giant virus possesses even part of this apparatus, normally reserved for cells, raises a major question. How close does it come to the cellular world? Imagine a simple scenario. An animal or plant cell usually defends itself against viruses through immune barriers or walls. Often, the virus infects, replicates, then destroys the cell. However, a megavirus, with a wide range of genes, some related to translation, could interfere more deeply with the host. It could introduce genetic fragments into the cell capable of interacting with the protein synthesis machinery, even redirecting the production of certain molecules. This disruption of essential cellular functions could accelerate evolution, because when the cell reproduces or releases its genetic content, this mixing introduces innovations into microbial ecosystems, pillars of terrestrial biodiversity. At this stage, let's discuss the concept of virophage. A virophage is a virus that parasitizes another virus, akin to the Sputnik discovered alongside mimivirus. When the megavirus enters a host cell, the virophage hijacks the machinery installed to produce its own particles, thereby reducing the productivity and virulence of the giant virus, sometimes even saving the infected host. This true molecular chess game also favors unexpected genetic transfers, thus generating novel genetic combinations that are potentially carriers of evolutionary innovations. To gauge the scale of these modular recombinations, imagine the genome as a set of building blocks. Genes assemble, disassemble, or reorganize themselves, altering cellular or viral development. The emergence of new genes, or the alteration of old ones, brings forth novel functions. In nature, these occasional events reconfigure the chain of life. Thus, a virus equipped with a gene-bypassing immunity can, through transfer and replication errors, pass it on to another virus or to a cell. The gene then quickly spreads, locally modifies viral predation, and ultimately influences the evolution of hosts, both fauna and flora. The close coexistence between megaviruses, virophages, and host cells has inspired an innovative idea, the revisited virus-first theory. According to this hypothesis, viruses, including megaviruses, might have appeared very early in the history of life, possibly even before certain cellular branches, or at least in parallel. They could have represented a primitive form of genetic material organization. To understand this scenario, let's recall what a capsid is. It's a protein shell that protects the genetic material. This primordial compartment would have offered viral genomes valuable stability in the chaotic environments of primordial Earth. These are marked by strong ultraviolet radiation, sudden temperature variations, meteoritic bombardments, and an atmosphere very different from today's. In such a context, a fragile genome, whether RNA or DNA, could hardly survive without protection. The capsid would have thus acted as a natural shield, allowing certain genetic sequences to persist, evolve, and, over time, become more complex. If we consider the capsid as a protected microenvironment, we can imagine that, in this refuge, fragments of genes gradually assembled to give rise to the first building blocks of cellular life. This hypothesis reverses the classical vision that viruses derive from more complex cellular organisms that have lost their autonomy. Perhaps some megaviruses are actually remnants of an ancient quasi-cellular lineage, or even a parallel evolution that contributed to the emergence of the modern biosphere. Exchanges between viral entities and protocells could have occurred, each sharing genetic sequences and replication strategies, thus weaving a complex evolutionary puzzle. The discovery, in some megaviruses, of genes involved in translation or DNA repair reinforces the idea that a virus-first scenario could represent a missing link in the tree of life. Megaviruses can act as true evolutionary accelerators. When a giant virus manages to manipulate, even partially, a cell's protein production, it alters its ability to respond to the environment, repair its DNA, or release new viral particles carrying genetic innovations. This influence goes beyond the microscopic world and extends to multicellular organisms that unknowingly host entire communities of microbes and viruses. In coral reefs, for instance, symbiotic protists are essential to coral health. If a megavirus disrupts their balance, the chain effect can lead to coral bleaching, alter the composition of microscopic algae, and ultimately affect the entire fauna and flora of the reef. A simple viral change can thus upset the whole trophic chain. Tropical forests, true reservoirs of life, could harbor megaviruses infecting soil, leaf, or root microbial communities. The co-presence of megaviruses and virophages promotes gene exchanges likely to generate new microbial strains. Equipped with novel degradation or metabolic capabilities, they can transform nutrient circulation in the forest ecosystem. Thus, the influence of these giant viruses extends beyond the microscopic sphere and affects, in the long term, the cycle of mineral elements, soil fertility, and plant health. A weakened flora alters the herbivore food chain, indirectly impacting insects, birds, and mammals. Far from being mere biological curiosities, megaviruses appear as potential regulators of biodiversity. Current research focuses on the structure and function of megavirus genes to shed light on their origins. The more we understand how these genes are transcribed into RNA and translated into proteins, the more we notice that some sequences are similar to those present in living cells. Their error correction mechanisms, complex enzyme production, and the sophistication of their internal structures suggest they could, under certain conditions, be more autonomous than previously imagined. This reignites the question: are these entities the last descendants of a primitive viral world, or the result of a gradual assembly of genes from various branches of life? It becomes clear why the virus-first theory is not unanimously accepted. Some researchers advocate a coevolution scenario, where larger viruses accumulated part of their genetic material through countless exchanges with various hosts over time. Their gigantism might stem from this ability to integrate useful sequences while remaining obligate parasites, opportunists capable of recycling their hosts' molecular innovations. For proponents of this approach, the important question is not which came first, but how the encounter between a virus and a host can accelerate genetic innovation. By analyzing the genomes of megaviruses, scientists have identified gene modules. These are sets of sequences working in concert to accomplish a specific biological task, such as replication or the construction of a cellular structure. When a megavirus infects a host, some of these blocks may integrate into its genome, or conversely, cellular modules may join that of the virus. This modular recombination generates great diversity, offering new capabilities: resistance to chemical stress, exploitation of a rare food resource, or development of an unprecedented immune defense. To concretely evaluate the influence of these interactions, researchers combine microbiology, biochemistry, ecology, and geochemistry. By analyzing samples of seawater or sediments, they detect genetic sequences specific to megaviruses, then observe how they vary according to salinity, temperature, or microscopic algae composition. The maps obtained often reveal a high concentration of megaviruses in areas rich in organic matter or algal blooms. These nutritious environments favor their encounter with potential hosts and other viruses. Unexpected symbiotic relationships are sometimes discovered. Microscopic fungi infected by megaviruses coexist with bacteria and archaea in a single sample. This proximity promotes genetic transfers and recombinations. The addition of a virophage further complicates the system. The host undergoes the attack of the giant virus, the virophage parasitizes the latter, and each tries to exploit the cellular machinery, disseminating DNA fragments. In just a few generations, new traits can emerge, and over millions of years, this mechanism could profoundly reshape life on Earth. Some researchers believe that megaviruses may have played a key role during major biological crises. Imagine a volcanic episode causing significant temperature variations, acidification, and changes in water salinity within a few days. Organisms stressed struggle to survive. A megavirus capable of exchanging gene modules could then promote the emergence of more resilient microbial variants while evolving itself. Under these conditions, forced competition and collaboration mingle, with megaviruses acting as gene distributors and strengthening life's resilience to extreme environments. This panorama blurs the once clear boundary between the viral and cellular worlds. The evolution of fauna, flora, and primitive microorganisms cannot be fully understood without considering the action of giant viruses. Far from being mere pathogens, they can rapidly and profoundly reorganize genetic networks. Our current view reveals a global ecosystem where viruses, cells, bacteria, archaea, plants, and animals co-evolve, sharing or disputing the same genes. With their large size and unusual genome, megaviruses offer an ideal field of study to uncover these subtle mechanisms. We dive between 3.5 and 2.5 billion years before our time. The planet then offered a radically different environment, but some fossilized traces reveal the activity of microbial communities. These marks, modest in appearance, are essential for understanding how life organized itself in a still hostile world. We will examine three major fossil biosignatures: the stromatolites of Warrawoona, the tubular microfossils of Barberton, and biogenic minerals, such as pyrite framboids or barite, enriched in sulfur-34 isotope. To begin, let us return to Warrawoona in Western Australia, a major geological site for the study of the earliest evidence of life. There are formations there that are 3,500,000,000 years old. At that time, the Earth was subjected to intense ultraviolet radiation due to the lack of a stable ozone layer, and the atmosphere contained much less oxygen than today. The temperatures, higher, fluctuated between 30 and 70 degrees Celsius. The oceans, with a different chemistry, rich in dissolved iron and potential nutrients, already housed microbial communities adapted to conditions hostile to most current life forms. These microorganisms, distant ancestors of bacteria and other cellular forms, sometimes formed microbial mats, thin layers made up of cells, filaments, and gelatinous materials. Over time, sediments and particles settled there, and other organisms settled, creating laminated, multi-layered structures. This type of structure is called a stromatolite, from the Greek for mat or layered rock. At Warrawoona, these superimposed formations reveal the repeated activity of living communities. Stromatolites exhibit concentric or undulating lamination visible in cross-section. Each layer marks a phase of microbial growth, followed by sedimentation. The accumulation of these alternations over thousands or millions of years sometimes forms massive structures, reaching several tens of centimeters. At Warrawoona, their origin has long been debated, microorganisms or mere geological phenomena. Biochemical analyses, notably of carbon isotopes, have finally confirmed their biological nature. Isotopes are variants of the same chemical element, differing by their number of neutrons. Thus, carbon-13 and carbon-12 are two forms of carbon. In inanimate matter, their ratio differs from that observed in living matter. Microorganisms using carbon for photosynthesis or other primitive processes favor carbon-12 because it is lighter. This depletion in carbon-13 is found in fossils. The isotopic analysis of Warrawoona's stromatolites revealed this signature, confirming their biological origin. These results were crucial for science, attesting to the existence of organized life on an Earth still young and the ability of microbial colonies to build durable structures. Imagine a shallow shoreline, bathed in warm waves, covered with bacterial mats, trapping and cementing mineral particles to slowly form these stacks. True reefs before their time, stromatolites hosted primitive ecosystems, microbial communities without skeletons or leaves. But already, they are capable of shaping and transforming their environment in the long term. Let us now look at the Barberton Greenstone Belt in South Africa, which contains some of the oldest terrestrial formations, about 3.2 billion years old. In these rocks, researchers have identified fine, tubular, sinuous, and organized structures reminiscent of biological filaments. They could be the remains of filamentous microorganisms capable of metabolizing sulfur or iron, or drawing their energy from other primitive chemical reactions. To understand how these tubes were identified and why they are considered biological in origin, one must examine modern analytical techniques, such as Raman spectroscopy. This method involves sending a laser beam on a sample. The molecules then return light slightly shifted in frequency. This shift reveals atomic bonds and allows distinguishing between organic and inorganic matter, even identifying the precise nature of the material. Moosbauer spectroscopy relies on the nuclear resonance of an element, most often iron, and measures the variations in its environment to determine the chemical or mineral structure of the rocks. A distribution of iron suggesting microbial influence rather than a geological deposit is a strong indication of past life. At Barberton, this method, combined with Raman spectroscopy, electron microscopy, and petrographic analysis, has shown that the tubules were very likely microbial in origin, and not the result of a purely physical process. From Warrawoona to Barberton, the observations reveal a microbial flourishing in the primitive oceans, adapted to various environments. Some thrived in warm waters, common during the Archean. Macroscopic fauna and flora were absent, but these microorganisms already formed entire ecosystems. Invisible to the naked eye, they left detectable signatures in the rock thanks to specialized analytical techniques. Among the clues confirming biological activity in these ancient rocks are certain minerals called biogenic. They are formed thanks to living organisms rather than by simple inorganic processes. This is the case for framboidal pyrite, a shiny sulfur iron mineral nicknamed fool's gold. Unlike regular crystalline forms resulting from abiotic processes, framboids have a globular aspect reminiscent of a raspberry, formed by the aggregation of spherical microcrystals. The framboidal configuration of pyrite is very often associated with the action of sulfate-reducing bacteria, microorganisms that derive their energy from transforming sulfur compounds. In other words, they breathe sulfur rather than oxygen, allowing them to live in environments devoid of free oxygen. The formation of framboidal pyrite, therefore, reflects the activity of these bacteria, which reduce sulfur and cause the joint precipitation of iron and sulfur as this mineral. Finding these framboids in very ancient rocks, especially when abundant, is a strong indication of the existence of specialized microbial ecosystems. Another emblematic biogenic mineral is barite, a barium sulfate sometimes enriched in sulfur-34. Like carbon, sulfur exists in several isotopes, including sulfur-32 and sulfur-34. Certain biological processes favor one of these isotopes, thus altering their ratio. In barite, a marked enrichment in sulfur-34 suggests that part of this element has been transformed by bacteria or other microorganisms involved in sulfur reactions. Thus, the barite present in these ancient rocks retains the chemical signature of microorganisms that once lived in seawater or coastal and hydrothermal areas. The study of isotopic distribution allows moving beyond simple visual observation to understand the essential role of these tiny beings in the formation of the mineral. It is, in a way, reading in the rock the imprint left by living communities. The idea of chemical signatures is essential. We cannot conclude the existence of ancient life based on a mere strange-looking rock. In the past, some hasty interpretations of mineral structures have been questioned. In-depth isotopic analyses allow these hypotheses to be validated or refuted by revealing the footprint of a stable fractionation typical of biological influence. These methods offer a true interpretive framework, like a palimpsest, where each layer reveals chemical markers of ancient ecosystems. The discovery of stromatolites, tubular microfossils, and biogenic minerals in rocks aged 3.5 to 2.5 billion years reveals an already living and diverse Archean biosphere. No lush forests or complex fauna, the microscopic world dominated, covering the rocks, altering the water's chemistry, and initiating the colonization of the terrestrial environment. In various environments, these microorganisms invented and perfected survival strategies passed on to their descendants. These fossil biosignatures remind us that life does not need oxygen to exist. In the Archean, its atmospheric level was minimal, and it took a long time before oxygenic photosynthesis transformed planetary chemistry. Metabolisms then relied on the chemistry of iron, sulfur, or hydrogen, exploiting niches near hydrothermal vents or sediments enriched by thermal or volcanic activity. What seemed like a hostile Earth actually offered multiple opportunities to microorganisms. The fact that these microorganisms left such clear imprints in the rock, lamination of stromatolites, barberton tubules, isotopic signatures in pyrite or barite, is remarkable. This offers an exceptional opportunity for research. This fossil record, much older than the plant or animal fossils of the Phanerozoic, requires patience, advanced analytical methods, and a solid interdisciplinary approach, blending geology, biology, chemistry, and physics to interpret these microscopic specimens. Imagine a wanderer on Earth 3.5 billion years ago, a landscape of dark rocks, sometimes reddened or yellowed by oxidation, bordered by seas ranging from green to ochre, depending on their composition. No fish, birds, or plants, only a world of invisible microbes proliferating on the ground or in shallow waters. These tiny life forms, true organizers of the biosphere, altered minerals, transformed the water, and shaped the sediments. Today, their fossilized traces provide geologists with a fascinating account of the beginnings of life. Terrestrial fossil traces illuminate the search for life elsewhere in the universe. Exobiologists seek to identify the signatures that extraterrestrial life might leave in the atmosphere of an exoplanet or in planetary rocks. The experience gained from analyzing the Warrawoona stromatolites, the Barberton microfossils, and measuring sulfur isotopes in sulfur-34, enriched barite guides the detection of life clues on Mars, icy moons, or exoplanets. If, as early as the Archean, terrestrial life produced a measurable isotopic fractionation, it is plausible that extraterrestrial organisms also alter their environment in a detectable manner. In summary, between 3.5 and 2.5 billion years before our era, microbial activity already left fossil signatures in the rock. In a world without ozone, in waters bubbling with primal chemistry, life demonstrated its prowess. Bacterial mats erecting mineral hills, altering planetary chemistry, and transforming the environment. Their goal was not to build monuments, but their accumulated presence over hundreds of millions of years has shaped the geological archive. To trace these ancient communities, well-preserved sedimentary rocks are first examined. It is discovered that microscopic life can leave tangible traces, even in very ancient geological contexts. Certainly, debates persist, but a convergence of converging evidence often strengthens the biological thesis. Sometimes, an unexpected detail in the morphology of a fossil, a chemical coloration, or the distribution of sulfur-34 in barite revives the discussion. This is where the interest of science lies, progressing through verifications, cross-checks, and productive controversies. Ultimately, fossil biosignatures dated to 3.5 to 2.5 billion years ago are among the most fascinating evidence of life's impact on a young Earth. These traces confirm that, even billions of years ago, the co-evolution of Earth and life was underway. Microbial activities, invisible in the short term, cumulatively transformed the chemistry of the air, oceans, and sediments. They paved the way for new metabolic strategies and increasingly sophisticated adaptations, leading to the emergence of complex organisms. It all starts here, in the silence of Archean rocks, where minerals and laminated strata tell of a distant world already deeply alive. Thus concludes our journey through fossil biosignatures that form the record of the early stages of life. This story engraved in mineral letters before macroscopic beings. With these vestiges, Earth offers us a famous chapter, an invitation to admire the creative power capable of transforming inert matter into a living world. In the ancient history of Earth, a crucial chapter hinges on great cataclysms and the resilience capacities deployed by the earliest living organisms. There were no forests, animals, or complex plants then, only microscopic communities, colonies of cells feeding on chemical or light energy. Despite their apparent simplicity, these life forms showed remarkable ingenuity in the face of ultraviolet radiation, asteroid impacts, or extreme cooling, turning the planet into an ice ball. We will explore these periods to understand how life, fragile yet tenacious, survived and evolved thanks to protective systems and adaptation strategies inherited from those distant times. At the dawn of Earth, the atmosphere had no protective ozone layer. Today, this barrier blocks most of the harmful ultraviolet radiation from the sun, but billions of years ago, living beings were directly exposed to it. These energetic electromagnetic waves can severely damage biological molecules, notably DNA, risking fragmentation or causing irreversible mutations when they struck primitive cells. To counter these threats, some microorganisms developed remarkable molecular repair mechanisms, including photolysis. This enzyme uses visible light to correct the lesions caused by ultraviolet rays. By binding to damaged DNA and utilizing certain less aggressive wavelengths, it restores the altered chemical structure. Thus, even exposed to harmful radiation, the first organisms could exploit a milder part of solar radiation to repair themselves. The emergence of photolysis was a crucial adaptation. Microorganisms capable of using it, as well as other similar systems, gained a clear evolutionary advantage. On primitive Earth, they had to survive in extreme environments, sometimes directly exposed to sunlight on rocks or in shallow basins. Combined with refuge behaviors, migrating into fissures or associating with protective minerals, photolysis formed a true living shield, well before the appearance of the ozone layer. Remarkable as it was, photolysis was not the only defense of primitive microorganisms. Some also produced ultraviolet-absorbing compounds, acting as protective pigments to limit damage before repair. The primitive atmosphere, rich in carbon dioxide, generated a greenhouse effect, maintaining high temperatures in air almost devoid of oxygen. These harsh but tolerable conditions for extremophilic microbes favored communities settled in temperate pockets or near hydrothermal sources, where photolysis played a key role against direct solar aggression. Aside from the constant threat of solar radiation, primitive Earth endured violent asteroid impacts. Massive celestial objects struck the planet, creating immense craters and reshaping the landscape over thousands of kilometers. Two emblematic sites testify to these events: Vredefort in southern Africa and Sudbury in Canada. Although life was then limited to microbial forms, the geological and climatic consequences of these bombardments may have had a lasting impact on the evolution of the biosphere. The Vredefort crater, one of the largest known on Earth, is estimated to be about 2 billion years old, a time when microorganisms were colonizing various environments. The impact of an asteroid several kilometers in size released colossal energy: local rock fusion, temperatures of several thousand degrees, ejecta, projected into the upper atmosphere and falling back in an incandescent rain. Without forests, the landscape remained mineral, punctuated by microbial mats. For this microscopic life, the impact was a cataclysm. Extreme heat, enormous pressures, and shock waves could eradicate many colonies, even those sheltered in the rock. Sudbury, formed around 1.8 billion years ago by a giant asteroid, created a vast basin still visible in Canada. As with Vredefort, the effects extended far beyond the impact zone. Massive projection of dust and gases obscured the atmosphere and reduced ground luminosity. This veil could sustainably lower temperature, disrupting the already limited primitive photosynthesis. The gases released, including various oxides, modified the atmospheric chemistry, influencing the formation of ozone and the concentration of carbon dioxide. Far from signaling the end of life, these impacts sometimes favored the spread of robust microorganisms. The ejected materials could carry colonies protected in small rock fragments resistant to extreme heat. Some hypotheses, still debated, even suggest expulsion beyond the atmosphere. The most resilient organisms, equipped with solid DNA repair mechanisms or reinforced membranes, recolonized the devastated areas as soon as conditions became tolerable again. This repopulation, on long timescales, illustrates the persistent dynamism of life in the face of geological crises. Through the craters of Vredefort and Sudbury, we read the echo of major cataclysms, but also the adaptability of an emerging biosphere. These impacts, while causing local extinctions, created new environments, released rare minerals, and perhaps favored the diversification of microbial lineages, exploiting these disturbed environments. Survival required resistance to thermal shocks, extreme pressure variations, and reduced light, preparing life, in the long term, for other planetary upheavals. In addition to celestial impacts and intense ultraviolet radiation, the early ages of Earth experienced extreme glacial episodes. During these phases, known as snowball Earth, the planet could have been almost entirely frozen, from the poles to the equatorial regions. Although their exact extent is debated, many geological clues, particularly deposits associated with glaciers, suggest that thick layers of ice covered continents and part of the surface oceans. When Earth was almost entirely covered in ice, the whiteness reflected most of the solar energy, maintaining the frozen state. Without vegetation or animals, the planet resembled an isolated white ball. This context seemed fatal, but life found refuge in volcanic and hydrothermal areas, generating favorable heat and chemical gradients. Even under a global ice cap, some volcanic zones pierced or thinned the ice, creating small pockets of liquid water. In these conditions, heat from eruptions or underwater hydrothermal circulation offered vital energy. Hydrothermal vents, releasing superheated water, could host robust microbial communities capable of exploiting chemical energy by oxidizing sulfur or other elements while escaping the freeze. The mineral-rich volcanic waters provided nutrients and stability. The pressure of the depths remained more constant than the extreme surface temperatures, often well below zero. The role of subglacial volcanic areas was essential. True refuges, they offered microbes thermal and chemical stability during glaciations. At the end of these episodes, the greenhouse effect, amplified by volcanic carbon dioxide and other geological sources, caused the ice caps to retreat. The protected ecosystems were then able to recolonize the liberated oceans and lands, expanding in more hospitable environments. As the ice receded, sunlight once again became accessible, reigniting primitive photosynthesis and other metabolic pathways. This energy promoted microbial growth and the acquisition of additional resources. The massive melting stirred the waters, dispersing these organisms on a global scale. Thus, even in its rudimentary form, life showed remarkable tenacity, surviving the Great Cold and exploiting the slightest bit of warmth and light to continue its evolution. Many scientists believe that it was in these protected niches that the mechanisms for freeze resistance were developed, such as the production of antifreeze molecules or the formation of protective layers around cells. These adaptations, born from glacial crises, would be the ancestors of an arsenal of strategies later enabling other forms of life to colonize extreme environments. Thus, Archean snowball glaciations were not just a trauma, but also acted as a catalyst for innovation, pushing microorganisms to invent new solutions to preserve their vital functions in the face of ice and lack of light. The ecological reconfiguration following the ice melt multiplied the available habitats, injecting new vigor into life. After the white chaos, the emergence of open waters and barren lands offered virgin spaces to conquer. Cold-resistant extremophile microorganisms able to endure stress spread across the world, passing on their genetic toolkit for resistance and cellular repair. Cumulatively, these Archean glaciations may have strengthened the robustness of the biosphere, endowed with defenses whose derived forms can still be found in today's fauna and flora. Ultimately, this period demonstrates that life had an exceptional adaptive potential even at its beginnings. Thus, the early Earth was not a static backdrop, but a vast laboratory of biological innovations. Early cataclysms and resistances wrote the first chapters of a story where each constraint called for a solution. These adaptive responses laid the foundations for future ecosystems. It's hard to imagine that today's fauna and flora descend from such elemental creatures that face such a hostile world. Yet, it is in this succession of extreme challenges and ingenious solutions that the persistence of life is rooted. Exploring these trials reminds us that evolution has never been a smooth process, but a constant struggle between the environment and the adaptive capacity of life, foreshadowing the countless battles it will continue to fight. As our gaze turns to the vastness of the cosmos, a question arises. Could life, as we know it on Earth, thrive elsewhere? In this final stage of our journey, we will delve into contemporary exobiology, exploring how the study of the atmospheric composition of certain planets and moons, combined with new remote observation technologies, fuels the hypothesis of extraterrestrial life. We will then broaden the reflection to alternative forms of life, potentially based on other chemical elements or solvents than those familiar on Earth. Finally, we will see how these discoveries and hypotheses, in return, illuminate our understanding of the emergence of life on our own planet. In the past few decades, the search for life beyond Earth has shifted from the realm of science fiction to become a genuine scientific quest. Exobiology, or astrobiology, studies the possibility of life elsewhere in the universe by combining chemistry, biology, geology, planetology, and astronomy. This intersection of disciplines helps understand how life could appear, organize, and persist in environments sometimes very different from those of our planet. Research relies in particular on identifying the conditions favorable to life and detecting revealing signs called biosignatures on the surface or in the atmosphere of nearby or distant celestial bodies. Among the most promising celestial bodies in our solar system, Mars holds a special place. Long considered arid and inert, the red planet has revealed, thanks to probes and exploratory robots, that it once harbored rivers, lakes, and perhaps a vast ocean. Even more surprising, recent measurements have detected variations of methane in its thin atmosphere. This unstable gas disappears quickly without a source of replenishment. On Earth, it mostly comes from living organisms, but also from certain geological processes. On Mars, this mystery remains unresolved. Unknown geological activity or subterranean life nestled in ancient water pockets. Future missions plan to drill into the Martian soil to analyze the isotopic composition of methane and search for complex organic molecules, possible signatures of biological activity. Around Saturn, the moon Enceladus has become a major target of exobiology. Long perceived as a simple block of ice, it has revealed, thanks to the Cassini probe, impressive geysers erupting from its poles, projecting into space water vapor mixed with mineral salts and carbon compounds. These plumes betray the presence of a global ocean beneath the icy crust, probably heated by internal activity and tidal forces. Hydrothermal vents could create conditions conducive to microbial life, comparable to those of primitive terrestrial hot springs. Enceladus offers a unique advantage. Indeed, its geysers allow sampling from space of this hidden ocean. Cassini analyses and future missions aim to refine this chemistry in hopes of finding direct evidence of an ecosystem. Among the most promising methods for detecting life at a distance is the spectral analysis of planetary atmospheres. When a planet transits in front of its star, the light passes through its atmosphere before reaching our telescopes, imprinting the chemical signature of its gases. The simultaneous detection of oxygen and methane would be a strong indicator. These two gases react quickly with each other, and their persistent presence implies active replenishment. On Earth, photosynthesis, respiration, and methanogenesis ensure this cycle. Observing such a pair on a distant planet could thus signal a biological process or, at minimum, an unknown source fueling these gases. This approach, called transit spectroscopy, today benefits from the increased capabilities of new terrestrial and space telescopes. Beyond gases, exobiology also explores colors as biosignatures. On Earth, many organisms produce pigments to capture light, protect against UV rays, or store energy. These colored molecules sometimes give green hues, like chlorophyll, or purple, like Bacteriorhodopsin. Thus, a planet covered with purple microorganisms could display a distinct light signature in its reflected spectrum. Some studies even estimate that early Earth could have shown a slight purple hue due to the abundance of proteins similar to Bacteriorhodopsin. If one day we detect, on an exoplanet in the habitable zone, such a colored spectral signal, it would be a strong clue prompting a closer examination of its chemistry and its variations over time. Still within this cosmic opening, modern science questions the range of forms that life could take. Our classic concepts are based on carbon chemistry, liquid water as a solvent, and the use of nutrients such as nitrogen, phosphorus, or sulfur. However, some researchers envision a radically different chemistry, adapted to extreme environments, even within our own solar system. The appearance of life relies on a network of chemical reactions, capable of replicating and persisting against thermodynamic constraints. Yet, the periodic table offers other candidates, such as silicon, which can form molecular chains, though less varied and more fragile than those of carbon. A silicon-based life might thus require a solvent other than water or particularly high temperatures to develop and sustain itself. In the same way, liquid water is considered the cornerstone of terrestrial life. Yet, on some distant worlds, liquids such as methane or ethane remain stable at low temperatures, like on Titan, Saturn's moon. There, a complete meteorological cycle relies on methane, evaporation, condensation, rains, lakes, and seas. In such a setting, an adapted biochemistry would need to use liquid methane as the main solvent, involving reactions and structures very different from those known on Earth. Ammonia, liquid under certain conditions, could also serve as a solvent for unprecedented life forms. These scenarios, still hypothetical, fuel the research question: how to build a cell without water or traditional carbons, but with other molecules adapted to extreme environments. All these speculations, although hypothetical, remind us of a key lesson. Avoid projecting our terrestrial models onto the entire cosmos. We know only one concrete example of life, the terrestrial biosphere. Thus, our definitions often rely on the idea that life requires water, exploits light, and relies on carbon. Yet, each discovery of extreme environments or new terrestrial organisms pushes us to rethink our certainties. Some microbes survive at over 100 degrees in ultra-acidic waters or under intense radiation, a proof of adaptability perhaps even broader than we imagine. This conceptual flexibility also sheds light on Earth's past. Studying extreme worlds pushes us to revisit our theories on the origin of life right here. Discovering a stable mixture of oxygen and methane on an exoplanet would almost certainly indicate a biological process, reigniting the question of the mechanisms that could have produced such a balance on early Earth. Conversely, finding on Mars or Enceladus an active organic chemistry, but without life, would confirm the delicate threshold separating prebiotic chemistry from true biology. It is important to point out that some space missions primarily search for traces left by life, rather than life itself. Even if extraterrestrial ecosystems existed, there is no guarantee they haven't disappeared long ago. The study of Martian geological layers, or Enceladus's plumes, thus aims to detect fossil indicators or molecular debris, indicating a biological past. This could be minerals altered by living processes, or even tiny typical amino acids. The drilling projects on Mars and sample collection around icy moons are therefore essential to confirm the possible existence of a vanished life. Such relics, even fragmentary, would have a major impact. They would show that life might be a common phenomenon in the universe and not an exception unique to Earth.
Everyone has gone home, but you still dance with me when the party is over. When the party is over.
This notion that life might manifest in multiple ways on other worlds brings us back to an almost philosophical question. What is life, really? Biologists have not yet found a universally accepted definition, each attempt leaving out a crucial criterion. Some favor the ability to reproduce, others the existence of an autonomous metabolism. Still others regard it as a system capable of self-maintenance through cyclical or autocatalytic chemical reactions. Exobiology even suggests that elsewhere in the universe, there might be systems capable of adapting and evolving without embodying most of the characteristics of terrestrial life. One day, we might identify in an extraterrestrial atmosphere or under an ice ocean a chemical organization unprecedented on Earth, yet which we would judge alive because of its internal functions and its ability to perpetuate itself. Such a conceptual revolution would also upend our understanding of terrestrial life's origins. We currently think it appeared through the gradual assembly of simple organic molecules and the formation of self-sustaining structures in a favorable liquid environment. but the discovery of life based on different chemical principles would open up other possibilities. Earth might not have been alone in experiencing a prebiotic scenario. Other competing chemistries might have existed here before disappearing or leaving a faint trace. Thus, the quest for extraterrestrial life reflects back on our own origins and questions our assumptions about the transition from chemistry to biology. With more sensitive instruments, advanced telescopes, and probes capable of reaching previously inaccessible terrains, research is advancing in the detection of signatures of life. On exoplanets, spectrometry refines atmospheric analysis, while in our solar system, missions collect samples of soil, ice, or vapor to determine whether the observed chemistry is geological or biological. Detecting an apparently trivial clue can overturn our knowledge. For instance, finding complex lipids or peptides in the plume of Enceladus would make it a priority target for exobiology. On Mars, abnormal seasonal variations of methane lead to comparisons with terrestrial archaea to verify a biological origin. Similarly, a terrestrial-sized exoplanet in the habitable zone with an atmosphere rich in oxygen and methane would trigger a race for confirmation. New generation space telescopes could then clarify these signals and test our hypotheses. This research also sheds light on our early history. If advanced organic chemistry can emerge on a sterile body in less than 1 billion years, it prompts a re-evaluation of the possible speed of life's appearance on Earth. Conversely, if key molecules, like primitive nucleic acids, form only in rare contexts, the origin of life could depend on an exceptional sequence of events. Each cosmic discovery and exobiological mission thus refines our understanding of the dawn of terrestrial life. This cosmic opening is not just a detour into the unknown. It acts as a mirror of our origins. The more we accumulate observations and test our theories in unexpected environments, the better we grasp the complexity of the laws that led to life on Earth. If life exists elsewhere, it may surprise us with its similarities or astound us with its differences. In either case, it will enrich our understanding of what life is and its possible manifestations. As major missions are being prepared, from the return of Martian samples to the exploration of icy moons, scientific excitement remains high. Proving the existence of another biology would be one of humanity's greatest intellectual revolutions. Perhaps life is neither rare nor unique to Earth, but widespread in the cosmos in 1,000 forms. By exploring these distant worlds, we might one day understand how everything started on our planet, our cradle, illuminated by the reflections of the universe.